Okay, so today we're going to put on our detective hats. We're about to see how a really powerful lab technique can crack a case that's literally life or death, tracing a dangerous disease right back to its source. And our whole investigation, it kicks off on a college campus where just one single diagnosis is about to cause a massive crisis. So picture this, a college campus, just like any other, but suddenly panic starts to creep in. A student has just been diagnosed with a really severe case of bacterial meningitis. We're talking a potentially fatal infection. And just like that, every single person she's been in contact with could be at risk. So here's the challenge for the health officials on the ground. They've got to figure out who else is infected, and they have to do it like yesterday to stop a full-blown outbreak. Now, the traditional method? It's a spinal tap to get cerebrospinal fluid. And yeah, it works, but it's also super invasive and painful. Trying to do that on dozens, maybe even hundreds of scared students, it's a total logistical and ethical nightmare. They desperately need a better tool. To find that tool, we actually have to look inside the human body itself. Because it turns out the most important clues are already being created by our own immune systems. Yeah, our bodies have their own team of highly specialized detectives. This image is great because it basically visualizes our internal security force. You've got the pathogen, that's the bad guy, like the meningitis bacteria. On its surface, it has these unique markers called antigens. Just think of those as the invader's fingerprints. And then you have the antibodies. These are the detectives our body custom builds to hunt for and grab onto those exact fingerprints. So the key takeaway here is this incredible specificity. An antigen is the unique molecular signature of a threat. And the antibody? It's the custom-made protein that can find and bind to only that one specific signature. It is a perfect lock-and-key system, which is exactly what scientists can take advantage of in the lab. And that brings us to the investigators' #1 tool for this case. We're going to take that natural detective system from inside the body and put it to work in a controlled lab environment. The tool is called ELISA. It stands for Enzyme-Linked Immunosorbent Assay, which I know is a total mouthful, but the concept behind it is just brilliant. It's a test designed to use those super-specific antibodies to hunt for an antigen in a patient's sample. And the best part? It uses a special enzyme that creates a visible color change to shout, "Hey, I found it." So how does this investigation actually work? It's basically a 5-step process. First, you lock down the fingerprint. You add the patient's fluid to a little well, and any meningitis antigens just stick to the surface. Step 2, send in the primary detectives. These are the antibodies that will only bind to that specific antigen. Step 3, bring in the reporting detectives. This is a second antibody that has an enzyme attached to it, and it only binds to the first antibody. Step 4, you add a colorless chemical called a substrate. And that leads to step 5, the big reveal. If that entire chain has linked up, the enzyme reacts with that substrate and the whole solution changes color. Color change means you've got a positive ID. No change, the patient is in the clear. But here is where ELISA gets even more powerful. It doesn't just tell us if the culprit is there, it can also tell us how much of it is there, and this is absolutely crucial for tracing the outbreak. You see, the test gives us 2 different kinds of results. You've got the qualitative result, which is a simple yes or no. Is the antigen there or not? But then you have the quantitative result, which answers the question, okay, but how much? By measuring how intense the color change is, we can figure out the actual concentration of the antigen in that sample. So how do we measure that concentration? With a really clever technique called serial dilution. It's a simple but super precise way of watering down a sample in steps, which lets you create a whole range of known concentrations to compare against. And this table shows you exactly how that works. It's pretty straightforward. You start with a known concentration, let's say 100 micrograms per milliliter. In the next well, you dilute it by 10, so now you've got 10. Dilute that one by 10, you get 1. You run the ELISA on all these known samples, and the different color intensities you get create a standard. It's like a color key that you can compare your patient's results to. Now, right here, this is the critical clue for our entire investigation. Think about it. A person who was infected more recently hasn't had as much time for the bacteria to multiply in their system. So a lower concentration of the antigen could signal a newer infection. This is the key to building our timeline. Okay, so the lab work is done, the results are in, it is time to pull all this evidence together and finally crack this case. Here it is, our evidence board. We can see Jill tested negative, which is a huge relief for her, but Sue, Marco, and Anthony all came back positive. Now look at the quantitative results. Marco has a low concentration, Sue's is high, and Anthony's is very high. The next step is just classic detective work. You take this hard data from the lab and you start cross-referencing it with patient histories. You know, who hangs out with whom? Who was at the same party last week? Who lives in the same dorm? And when you put it all together, the picture becomes crystal clear, leading all the way back to patient 1. Anthony, with the highest antigen level, has been infected the longest. He likely passed it to Sue, and she then passed it to Marco, who has the lowest level because he's the most recent infection. The ELISA test didn't just find the 6 students. It gave us the timeline, letting us trace the whole outbreak back to a single person. So we solved the campus mystery. But, this is so much more than just a clever story. This technology has some really profound real-world implications. Let's just quickly recap the big takeaways here. ELISA is an incredibly powerful and specific tool for diagnosing diseases. It tells us not only if someone is sick, but it also gives us major clues about how long they've been sick. And that combination of data is precisely what public health officials need to track, contain, and ultimately stop dangerous diseases from spreading. And its applications go way, way beyond meningitis outbreaks. I mean, get this. The basic technology behind ELISA is the exact same principle used in every single at-home pregnancy test. Instead of looking for a bacterial antigen, those tests use antibodies to look for the pregnancy hormone HCG. It's a perfect example of how this powerful lab tool is used literally every single day. So, we've seen how ELISA acts like a detective, solving these mysteries after an outbreak has already started. And that kind of leaves us with a final big question. With this kind of speed and accuracy, could this same technology, or maybe something like it, be used not just to react to diseases, but to proactively monitor and maybe even predict them before they ever get the chance to spread?